Welcome to EKGs Explained Clearly. By the end of this course, you'll have all the skills needed to confidently interpret EKGs in a systematic way. You'll understand the electrical activity represented on EKG paper that allows this process to happen, the heart to beat and cycle blood through our lungs and bodies. This is the final product. Whether or not you have experience with EKG interpretation, I think you'll find this series of videos very useful. If you understand the key foundations of how the heart works, it'll be much easier to learn and remember the nuances involved with the EKG that we'll cover later in this course. So we'll start with the anatomy and physiology of the heart, depolarization, repolarization, and then on to leads and specifics about EKG paper and how a tracing is captured. Next are specifics on EKG tracing, the P wave, the QRS complexes, the QT interval, the R to R interval, etc. We'll cover the impact of our nervous system and neurotransmitters on the heart, then on to rate, rhythm, axis, escape rhythms, PVC, bigeminy, tachyarrhythmias, ventricular tachycardia, and the key differences between ventricular tachycardia and paroxysmal supraventricular tachycardia with aberrancy, the QTC, and the potentially life-threatening torsades de pointes, atrial fibrillation and flutter, widened QRS complexes, and the various types of heart blocks, using precordial leads to hone in on certain areas of the heart, atrial enlargements and ventricular hypertrophies, acute coronary syndromes, myocardial infarction, and pericarditis. We'll talk about Q waves, ST segment and T wave changes that we see with ischemia, bundle branch blocks and fascicular blocks. And then at the end, we'll put it all together and teach you how to systematically read an EKG. We'll go through a normal one first, and then onto a variety of abnormal EKGs like sinus tachycardia, atrial fibrillation, multifocal atrial tachycardia, first, second, and third degree heart blocks, atrial enlargement, ventricular hypertrophies, hyperkalemia, rare anomalies, and many, many more abnormal EKGs. So let's begin with anatomy. This is an animation here of the heart beating, and there's a number of things that I want you to see so you'll understand this when we talk about the conduction system of the heart and the EKG. So the EKG, as you know, is a way of looking at the electrical activity of the heart. And it's really the electrical activity of the heart that is responsible for what we're seeing here right now. Just for those that don't know the anatomy of the heart, we've got the right ventricle here on this side. We've got the left ventricle here on this side. And these areas up here are the atria. So this is the right atrium here, and the left atrium is over here. Interestingly here, what we've got is the tricuspid valve. This is a valve that allows blood to flow in from the right atrium into the right ventricle and then gets pumped out through the pulmonic artery. And this is the pulmonic valve here, the semilunar valves, as you can see. And then what you can't really see very well is the aortic valve, which is here at the top, and this is where the left ventricle is pumping out into there. Now, one of the things that I want you to notice first off is if you look carefully, you'll see that the right atrium here beats before the right ventricle. Right atrium, right before the right ventricle. And you'll also notice similarly that the left atrium beats right before the left ventricle. Now, this is because of the anatomy of the conduction system. There is an electronic conduction system in the heart that is not made up of nerves, but instead of modified, specialized myocardiocytes. Okay? Up here is the sinoatrial node, which, because of its relatively quick activity, is the pacemaker for the whole heart. It then goes to the atrioventricular node, which is here, and then goes down this conduction system here called the His-Purkinje system. And as you can see, it goes up and it basically conducts the electric signal to the myocardium. Now, we're going to talk more about this in detail, but I wanted you to notice that it's this conduction system that basically makes this movement that we're seeing right here like a perfectly well-timed symphony. Depends on the analogy that you want to use. There's several different analogies that you can use. The conductor that is conducting a symphony, and you want to make sure that the violins and the cellos and the flutes and the viola and the timpani, they're all acting all at the same time so you get the maximum effect. In other words, everything is working in concert. The other way of looking at it is a little bit more destructive. You know that if people are trying to demolish a building, They will have certain areas of the building that are drilled out and weakened with sticks of dynamite. And when they activate that dynamite, it has to be activated in just the right order, in just the right fashion, so that the building comes down right on top of its footprint. Well, this is kind of what's going on here with the heart. This electric conduction system, the sinoatrial, the atrioventricular, and the his-Purkinje system, is activated in such a way that the electrical conduction goes and stimulates the heart in concert. So the electrical conduction starts up here and then goes to the ventricle. The reason for that is so that the atria contract right before the ventricle. The purpose of that is to get the blood from the atria down into the ventricle to increase the size of the ventricle right before the ventricles contract. That causes an increase in preload and allows your cardiac output to be improved. That's happening on both sides. So you can see that the atria are contracting first, and then the signal is placed down here to the his-Purkinje system. It travels down very quickly down this his-Purkinje system and then up here into the myocardium. So the effect is that essentially the myocardium is contracting all at the same time. And you want that to happen. Instead of having contraction occurring from the top down, you want contraction occurring all at the same time. Because of this very unique conduction system, this is going to look very unusual, or look very unique, I should say, on an EKG. What is an EKG? It's a way of measuring current. You know that when you take a battery and you place it on a voltmeter, if you put the positive end to the positive meter and the negative end to the negative meter, you're going to move the dial up so that you see that there is a difference in the voltage. When that voltage starts to move, that is when you have an electric current. And so what an EKG does is it sees how is this depolarization, or this movement of positive charges that's going down the his-Purkinje system, how does it look from the electric vision, if you will? In other words, if I had an ultrasound machine, I could see if there is fluid or fluid moving on the ultrasound. What an EKG does is it does exactly the same thing, except instead of fluid moving and objects that you could see, you're seeing electricity moving. And so that is what we're going to look at today. Atria contract first, then ventricles contract second, but they're contracting in concert. And then what happens is everything resets back to normal and you have the same contraction happening again. The next series that we're going to talk about is we're going to break things down to the smallest level, and then we're going to build it up so that we can finally see how we get back to this point again. Okay, so zooming in a little bit more now in terms of where we were, let's pull away all the muscular activity here and let's look specifically at this conduction system. I want to be very clear. The thing that you've got to understand that these are not nerves, okay? These are modified myocardial cells that conduct electricity very quickly, and we'll talk about how that happens. So the first one that you've got to know is the sinoatrial node. It is at the very top, and the reason why it is there and it is the pacemaker is because its intrinsic activity is the fastest. And since it is the fastest, it's going to cause depolarization to occur all the way down, and everything else is going to have to be in concert. So this is kind of the conductor of the concert. Now there are different pathways to get to the AV node. You can see here that the SA node conducts to the AV node. Now the AV node is right here, and it kind of holds things up. It kind of delays things. We'll talk about that in another lecture. So that what you have here is you have the sinoatrial node saying, okay, it's time to contract, and we'll talk about how that happens. And the atria contract, and then the electrical conduction gets down to here and it holds up. What that effectively allows the heart to do is to pump blood from the atria down into the ventricles and allows it the time to pump it. Remember, this happens so quickly, SA node to AV node. And then when the blood is in the ventricle, the AV node then conducts this depolarization that's occurring into the his bundle system, and it travels down and around so quickly, so very quickly, that essentially the entire myocardium that this innervates, okay, or that it penetrates, basically depolarizes almost all at the same time. So that again is sort of the map of this cardiac conduction system. And really it's this cardiac conduction system that allows the depolarization to occur in such a nice and timed way. Now, on an EKG, when we're actually looking at the electrical depolarization, are we actually seeing the electrical movement down this conduction system? The answer is no. This is such a small amount of electricity that it's almost imperceptible on an EKG. What is it that we're actually measuring? What we're measuring is the depolarization of the muscle. The depolarization of the muscle is very large in amplitude, and that is what we are picking up on the EKG. Kind of like if we are demolishing a building, what do we see on the video? Do we actually see the electricity going into the wires to the dynamite? No, we're actually seeing the dynamite blow up and the building coming down. That's, in other words, what we're seeing on the EKG. Even though there is electrical conduction going down this, what we're seeing electrically on an EKG is the depolarization of the actual muscle cells. Okay? So just be aware of that. Okay, so to get a little bit more of an insight about what's going on, let's go down to the microscopic level. And I want to show you a myocyte here. Now, the thing that you should know about a myocyte is just like any other skeletal or smooth muscle cell, it has a collection in it called the sarcoplasmic reticulum. We'll call that SR. And the SR is full of calcium. And the reason why it's full of calcium is because this calcium can be released into the cell upon depolarization of the cell, and we'll talk about that. And that's going to cause contraction of the striated muscle. So we'll just put a bunch of muscle cells here that's going to contract. Okay? And if you were to look histologically at the areas of the myofibrils that are in cells, you'll see how muscle cells work. But basically, calcium is what's going to activate them to contract. And they're made up of troponin and all of these other things that we won't get into. The key here, though, is the sarcoplasmic reticulum causes calcium to be released, and that's what triggers it. The question is what triggers the sarcoplasmic reticulum? Well, what happens is, as you may know, on the cell surface of these cells, you have a sodium-potassium pump. And the purpose of that is to pump sodium out of the cell. And as you know, sodium has a +1 charge. And at the same time, pump in potassium. And potassium has a +1 charge. But it pumps out 3 sodium for every 2 potassium. And so what ends up happening is there's a very high concentration of sodium, okay, 1+, outside the cell. And there's a very high concentration of potassium inside the cell. Now, as it turns out, the cell is very impermeable to sodium. So sodium stays very high outside of the cell. A way of thinking about it is because 3 are going out and 2 are going in, there is a negative charge on the inside of the cell. Okay? Negative charge on the inside of the cell. And there are positive charges, if you will, on the outside of the cell. We'll ignore those for now because the outside of the cell is very, very large and the inside of the cell is very, very small. So as a result of that, there's a far more negative charge that we see here. Now, the other thing that's important to know is that the cell, while it is very impermeable to sodium, it is very permeable to potassium. And as a result of that, potassium leaks out somewhat. Now, as it leaks out, it's losing a positive charge, so that even makes it more negative. And as a result, you have a very negative resting potential on the inside of the cell. Now, with that basis of history, let us go forward here in the next lecture and talk about why this interior negative charge is so important to the electrical conduction that we're going to talk about here in our EKG course. Thanks for joining us.